you can see that you can build these long filaments by successively adding the actin subunits on one and sometimes both ends of these filaments. And you can build pretty long structures this way. But I think that one of the most interesting features of the actin cytoskeleton is that it can build these tree-like branched structures when the long filaments of actin are joined together at a quite precise angle of about 70 degrees. And if you look at the front edge of a crawling cell, a lot of the action is happening at the front of the cell, which is called the leading edge. It is here where there is a lot of building and destruction going on of these actin trees. We can see this better in this diagram. So this is the front of the cell, the leading edge. And the new actin-based trees are continuously constructed at the front end. And this is what is pushing the front of the cell forward. You can appreciate how dense these structures are in this electron micrograph. Look at this. It is full of these actin-branched structures. At the other end of this arrangement, the filaments are continuously destroyed, and these subunits are then reused. They are brought forward, charged with energy again, and participate in the construction of new trees. And so I want to show you a couple of videos here in which you can see the leading edge of the cells in action. Here, actin is labeled with a fluorescent protein, and you can see its high level of concentration on these edges of the cell. And this is exactly where this constant rearrangement of the cytoskeleton is carried out. Now, let's ask a question. If you look at these motions, they may seem random at first. But let me show you another video of the slime mold amoeba dictyostelium that we saw a single cell performing early on. Now we have a group of cells here crawling to the source of attractant which is coming out of this capillary. You can see that the cells reorient themselves. They can somehow sense the presence of the signal there. And they will eventually aggregate here and form a large cellular assembly, as you can see. How does this happen? This is one of the main questions that we work on in the lab. So the general signaling pathway is represented here. Usually, the cells will sense the presence of extracellular signal molecules coming from the outside. They can be sent by neighboring cells, or sometimes they originate in the environment. These extracellular signal molecules interact with receptor proteins located on the surface of the cells. This event initiates a whole cascade of further events inside the cell that is collectively called signal transduction. This means that the signal is converted from one form into another through successive relay stages and mechanisms until finally, in our case, it arrives on the regulation of cytoskeletal proteins, and this can result in altering of the cell shape and generating motion. Now, these signaling pathways, as I mentioned to you, are of great interest to us because they establish the differences between the cells and they underlie a lot of the changes that occur during development. Now, so we have a pretty good idea now how these cells orient themselves to move. First of all, they have to perform directional sensing. They have to know where the signal is coming from. And these sensory systems at the level of single cells are extremely sensitive. The cells can detect a 2% difference of the chemical at the front versus the back. This resets cellular polarity, and so the actin gets concentrated at the front of the cell closest to the source. That causes the formation of a pseudopod by making these actin trees and finally results in movement. This is all good, but this occurs at the level of single cells. How do we then perform the movement of large cellular assemblies in the in the embryo and generate even more power and force. Here, we have to introduce another molecule, myosin, that can walk along the actin filaments, as shown here. So myosin has special head domains that move to the plus end of the actin filaments. And another interesting feature of myosin is that they can associate with themselves to form these bipolar structures in which you will have heads oriented in two different directions, and so one side tries to crawl this way, the other side tries to crawl to the left, and this results in relative movement of actin relative to one another. And so we now have a pretty good idea of the minute molecular events that happen during the movement of myosin along actin. And this cycle is shown here. At each step, the head of a myosin molecule will consume one molecule of energy in the form of ATP. And so here, when ATP is gained here, the myosin head is released from actin. It is ready for next motion. In the second step, this ATP is degraded and the energy is utilized. And so the head is cocked for the next step to interact with the second actin molecule. Finally, the head attaches itself to the actin and generates the power stroke to move the rest of the molecule forward. I have a great analogy for this mechanism here for you in competitive rowing. And if you notice, there are these terms here that are borrowed from that sport. So you can see, if you think of an oar as a myosin head, in the first step you extract it, you get it out of the water, then you recover it, so you prepare for the next stroke. You put it in the water, this is called catch, and then you perform a drive. This generates the power stroke. And so much like the rower here on the boat, myosin propels itself forward along actin. And this is all shown here nicely to you in this video representation. You can see that this is ATP gained and destroyed in the process at each of the steps. And this explosion is meant to show how energy is created and utilized in the process. All right. So this is a tiny molecule moving along one filament. But how do we move large things around? And the secret is in the large numbers. So myosin can associate into these bundles that are comprised of hundreds of these molecules. And moreover, they interact with bundles of actin, which are embedded in these vertical structures shown in blue. This segment is called a sarcomere, and it's a functional unit of our muscles. And when multiple myosin heads walk towards one side here, they're connected with their tails, and these heads try to pull in the other direction. But because they're stationary themselves, what happens is that instead these two vertical plates move closer to one another. And this is the basis of muscle contractility. Now, in order to build real muscles, you have to repeat this many times, both lengthwise and, importantly, widthwise. And that's how you get strong forces to contract. And as long as you have two proper attachment points, the muscle can contract. All right. Let's move back into the individual cells, because we want to see what happens in the embryo, not just in the biceps of Arnold. So the defining feature of actin-myosin contractility is very nicely represented in this structure called the contractile ring. During the division of a cell into two daughter cells, the final step is the separation of cells from one another. And this structure is built that contains a ring of actin and a ring of myosin around the place where the cells have to pinch away from one another. And as you can see in this movie, taken from the Drosophila embryo now, we're back to our favorite system. Myosin is labeled in green here, and you can see as the cells finish the process of cell division, there is this contractile ring that forms and finally dissipates. And the cells are now dividing. So actin and myosin play a key role in this process. So what have we seen in the second part here? We have seen that the cells can propel their edge forward by building the trees made of actin, an important molecule and filament. Cells can reorganize the cytoskeleton in response to signals from the outside. They can sense the signal coming in and appropriately send pseudopodia in the right direction. And a very large contractile power can be generated by adding a walking motor, myosin, that can walk along actin. So this is the end of part 2.